Okay, welcome back everybody from the coffee break. So we managed most of the plans that we had here. So we can't see Denis, but we can hear him, and we have his transparencies here. Denis is live in Oxford. Is that right? Are you in your lab or in your office?
Indeed, sir, yes.
Excellent. Okay, unfortunately he can't be here, but at least he can be here via the internet, and he will give his presentation on role of stochasticity in biological communication processes. And I'm very happy that you're here. So there are many people who are very interested in what you have to say. So Denis, all yours.
Okay, thank you very much. And first, let me say that I am very sorry not to be at the meeting. I've read many of the abstract, and it's clear to me this is a meeting I should have been present at in person. Sadly, I had an accident just a few weeks ago and still recovering from that. So, role of stochasticity in biological communication processes. If we go to the next slide, we will see the structure of my presentation. I will first of all do a little bit of reminding of history of some of the computational work done in relation to membranes and their processes in the context of unraveling complexity and dealing with downward causation. I will then use those models to look at the interpretation of knockouts, and then I will shift to the real focus of this talk, which is the role of stochasticity, and ask the question, is it just noise, or is it, by contrast, the harnessing of stochasticity to create function? So if we go to the next slide, I think there's just highlights in red that we're going to deal now with unraveling complexity. And the next slide, you would need to press buttons just to slowly go through the animations. It really doesn't matter, I will keep up. What this is doing for us is demonstrating all the various levels that computational biologists have been trying to reconstruct mathematically and how they may interconnect. And I'm showing an upward arrow to begin with, but I'm now going to put some downward arrows because I want to emphasize the fact that those lower levels are influenced enormously by the organization of higher levels. So if we now press the button progressively, we've got feedbacks down to triggering cell signaling. We've got systems level control of gene expression, which is the next one, I think. You can just keep pressing the button and it will go down, down, down. It really doesn't matter how often you go down. There we go. And one key thing you'll notice is that if you just go back, if that's possible, I've actually reversed the bottom little arrow to emphasize that a lot of the forms of downward causation do influence not only gene expression, but also gene sequences. So if we now go on to look at how we actually do that in practice in my own field, which is the membranes of the heart, we can now, next slide, look at, and again it's just a matter of pressing the buttons as quickly as possible. What we do, of course, is to put equations for all of the processes I'm illustrating into a cell membrane and cell structure model of varying complexity. And I'm now going to use one of those models to illustrate the process of downward causation in cellular systems. That is the way a parameter of the whole cell, in the case of excitation, it's voltage. But I've noticed that your symposium, your workshop, is actually emphasizing that there's much more to membranes than their electrical properties. I will hope to learn more about all of that. But anyway, let's go to the next slide, and it will animate to begin with on its own, I think, with the first, that's it, excellent. This is simply showing two of those ion channels, a potassium channel and a calcium channel, interacting to produce the electrical rhythm in the pacemaker region of the heart. All of this is based, of course, on detailed experimental information. Now what I'm going to do with the next animation is just to, there we go, I'm just going to hold the membrane voltage. And what we notice is that the oscillatory behavior of the ion channels completely disappears. And that is, of course, because we've broken the link in what's sometimes called the Hodgkin cycle, the influence of the electrical potential on the molecular mechanisms of the ion channels. And you notice that what that also does is to bring out the role of yet another channel. If we now put the link back on again, that's the next animation, there we go, and we will now see what happens if a drug influences that component that reappeared, that's the IF component. If we now press the next animation, we will see its acceleration by adrenaline. There we go. So what we're seeing here is clearly a form of what is sometimes called downward causation. If we go to the next slide, this was first, of course, emphasized by Alan Hodgkin during his work with Andrew Huxley on the electrical excitation of nerve. And so it's called sometimes the Hodgkin cycle. So if we now go to the next slide, this will highlight that I'm now going to use that model to look at the interpretation of knockouts. Before I go to the next, I'll stay with this slide for a moment because I want to explain what I'm going to say and show. When I first did these computations on the basis of some experimental data that I got many years ago on the potassium channels in pacemaking heart cells, I was able to reconstruct pacemaker activity with just very few channels indeed. At that time, it's about 1960, I had simply 2 potassium channels, which is what I'd done experiments on, and a sodium channel similar to that of Hodgkin and Huxley, and I was able to clearly show pacemaker rhythm emerging from that. What we subsequently found, as the slide earlier showed, is that there are very many other channels. Now, one interpretation of why nature has, during evolution, done that when it could have managed with just 3 is simply that the presence of just 3 channels, while it will produce pacemaker rhythm, is not robust. It's very fragile. The consequence of all the various other channels that have been discovered since and how they interact is that the system becomes very robust. And the next slide will show that in terms of, first of all, showing the rhythm again. It's the same as in the previous slide. And now we will see, as I progressively knock out fractions of the sodium channel there, what happens is that instead of getting a massive change of frequency, there's only a very modest change of frequency, in fact about 10%, even when the sodium mechanism there is completely abolished or knocked out. Now, that's in one sense very reassuring. It means that our pacemaker rhythm in the heart is a very robust process. But of course it creates quite a problem for interpreting gene knockouts, because if something that was contributing 80% of functionality, as that channel was doing at the beginning, and only shows a 10% change in frequency, then we have problems with interpreting gene knockouts when you come to look at overall function. And that's apparently quite general. The next slide will show the reasons for that. This is from a paper I wrote with Peter Cole, Edmund Crampin, and Alice Quinn some years ago, simply emphasizing that the way in which DNA, as a template for the proteins, communicates its effects to the phenotype is, of course, through the biological networks. And if we go to the next animation, we can see that, I hope the next animation, there it goes, yes, a single knockout may not go through. A double knockout may be necessary in order to get an effect at the phenotype level. Now, that's certainly what we find in pacemaker rhythm in the case of the heart. It's very robust. You would probably need at least 2 knockouts to get a complete abolition of rhythm, and maybe more than that. Nobody's yet done that, so I don't know the answer to that question. But the question does arise as a general one in relation to evolution, which is how extensive is this kind of genetic buffering, that is, the ability of the networks to manage even when a major component has been knocked out. Well, it looks as though it's pretty general. If we go to the next slide, this is from the work of Hillenmeyer and his colleagues just about 10 years ago, looking at the unicellular organism yeast, and looking at knockouts of many of the 6,000 genes in yeast. Each one of them, on being knocked out, was tested for how much effect that had on the phenotype in terms of the metabolism of yeast and its reproductive rate, and the answer is in the next animation, which is that 80% of the knockouts are actually silent. Now, that certainly does not mean that they're not functional. On the contrary, when you stress the organism with metabolic stress, meaning that it has difficulty getting some of the metabolites it requires, then you can show that many of those knockouts will have an effect. So it's another example of strong genetic buffering, and one can see then the reason for that. It means, of course, that environmental changes, particularly ones that influence the need for certain metabolites, may be resisted. So this leads us to the next slide, which is just highlighting where I summed all this up in an article for the Royal Society journal, Philosophical Transactions of the Royal Society, just about 10 years ago. I call it the solution to the genetic differential effect problem, but I think it's also a challenge for membrane biophysicists and physiologists, which is that can we, with suitable reverse engineering of good models, can we do what is the equivalent of what I did in the case of the heart, which is to find out the contributions of different components in circumstances where genetic buffering is very strong. So if we now go to the next slide, this will highlight that I'm now going to move on to the role of stochasticity and ask the question, is it just noise? But before I go to the slides that deal with that part of the presentation, again I would like to orientate people a little bit. People generally, I think, have thought that stochasticity, particularly in genetic components, that is mutations, random mutations of DNA, and perhaps also stochasticity in relation to ion channels in membranes, that this might be just looked at as just noise. I'm going to argue that it's much more than that. But I'm going to first emphasize how we got to the idea that it is just noise. So we go to the next slide, which asks a question, which is how did the system's approach to complexity become sidelined in the second half of the 20th century? And I think the answer is that we had an oversimplified view of the logic of living systems. I've used this mantra myself. From molecules to man. Now, the point there, of course, is that the prepositions, it's apparently a one-way process. And my problem with this is that I don't think this can be true. Molecules as such are not, after all, alive. So let's have a look at the origin of this mantra, which we've all tended to use. That's the next slide. And I think the origin of the idea, well, you can actually trace it back all the way to René Descartes, a philosopher in around 1660. But I think the person who really strongly influenced molecular biology as it developed during the 20th century was the famous physicist and quantum mechanics pioneer Erwin Schrödinger, and with various other predecessors. I've mentioned Descartes already, and Pierre-Simon Laplace would also be an example of a predecessor. Now, what did Erwin Schrödinger do that gave rise to the view that stochasticity is just noise? Well, let's go to the next slide. He gave a series of lectures in 1943 at the Dublin Institute of Advanced Studies, and he argued in that book very cogently. It's an excellent book, and I'd recommend reading it if you've not managed to do so. But he argued that there was a fundamental difference between physics and biology, that physics was order from disorder. At the molecular level, of course, you've got the movement, the stochastic movement of molecules, various processes going from quantum mechanical to thermal motion and many other processes that create disorder. But at a higher level, such as the whole cell, you can use thermodynamic approaches, which of course are much more smooth, and that's because of course you've got the law of large numbers, you've got all the various random motions, as it were, balancing each other out. Now, he then argued that by contrast, biology has to be order from order, and that's because, as I see it, as I read Schrödinger's book, he argued first that the genetic material can be compared to a crystal. He described it, I think, very accurately as a non-periodic crystal. And I think if you think of a polymer, a linear polymer like DNA, as a kind of crystal, then okay, it is non-periodic. That's how it contains information, of course, and can form templates for the creation of RNAs and proteins. Now, I think, and this is speculation on my part, crystals, of course, grow in a rather determinate way. Once you've got a seed for the crystal, then all the other molecules, as the crystal grows, determinately fit into the structure of the crystal as it grows. And I think he thought that, therefore, somehow or another, the copying of that crystal, what we now know, of course, to be DNA, was therefore determinate. Now, he struggles in the book with that concept. If you read, I think it's page 101, he says, well, first, this seems utterly absurd, and I think he's absolutely right. It would be absurd to imagine that biological systems, unlike physical systems, can be immune, as it were, to thermal and other forms of stochasticity. And indeed, they're not immune to such forms of stochasticity. A very simple way of explaining that is that, as we all know, the copying rate for DNA as it is first copied is actually quite high. It's around 1 in 10 to the 4 base pairs going wrong in the initial copying. But of course, we also know that the cell comes in with a whole army of protein mechanisms that correct that error rate and produce it or reduce it down to only 1 in about 10 to the 10, which means that a whole genome can be copied almost perfectly. So he was right about the idea that the passage of the information onto the next cell is highly accurate, but not that it was because it is or has a crystal-like determinant property. It's because the cell does all the error correction. So those are the reasons why, as we go to the next slide, I've highlighted 2 books there, one of my own and another of James Shapiro in Chicago, a biochemist. Both of us take the view that biology also largely, I want to emphasize that, creates order from disorder. The reason for saying largely is, of course, we know that there are various forms of disease states that can be attributed to a single mutation or change in a single gene. But of course, they are, in terms of the view of a medical scientist, the rare processes of disease that give rise to that. They are, if you go back to the Hillenmeyer type experiment, showing 80% of knockouts being silent, they're in the 20%, as it were, the ones that are not silent, even as a single knockout. So, what I want to do now, as I develop the last part of the lecture, is to look at stochasticity as a key feature of gene expression and also as a key feature of functionality. So if we go to the next slide, this is from a very important set of experiments done by Zhi Huang and his colleagues, looking at the random variations in different cells in a cell culture in regard to a particular protein. It doesn't really matter which protein you take, they all show stochastic variation in gene expression levels in the different cells. That's certainly true in the heart, the area that I work on. Various cells in heart structure show different degrees, very different degrees of expression levels of the various ion channels. Now, what I want to emphasize here, if we now press the button for the animation of this slide, just how big the stochasticity can be. In this particular case, with a cell adhesion protein that's been looked at here, the range is 1,000-fold. Now, you won't find the same range for every protein, but you'll certainly find stochasticity. Moreover, interestingly, and this is shown in the next slide, it is an attractor of the population, not of individual cells. What Huang and his colleagues did here, I think, was beautiful. They cloned either from the large level expressors in the case of a monomodal distribution, or from the higher expression group amongst the bimodal distributions that they occasionally found. And what you find in the progeny, if you do that, is that initially, for a day or two, you find the progeny express almost exactly the range of expression profiles shown by the parent cells from which they were cloned. But about 5 days later, you're back to the original distribution. Now, I've thought a lot about this experiment, and I think it's very difficult to resist the interpretation that what's going on here is that there is an attractor at the population level which is managing somehow or another to resurrect itself, even if you clone from the high expressors. So, if we go to my next slide, my conclusion from this part of the talk is that forms of stochasticity in gene expression are what we can call multicellular attractors. And then a big question, which is going to be the question I'm going to deal with in the last part of the talk, can that stochasticity be used by organisms functionally? And I think the answer is yes, it can, because it provides a range of expression to enable rapid response to stress. Now, it raises two questions. How widespread is that functionality, and how on earth do cells feed that information, which is sensed at the membranes, of course, with their various receptors and so on at the surface? How does it feed that information to influence the genome? Well, there are many experiments that show the details of how that can be done. The next slide I've taken from some beautiful experiments by one of my former collaborators in the early days when I was working on heart potassium channels. This is Dick Chen, who now works on the nervous system in New York University. And this is taken from the paper that's quoted at the bottom of the slide. What they were looking at was calcium entry at the surface membrane through calcium channels, leading to accumulation of calcium in a very local microdomain of the cell. Very tiny indeed. It's well known that these microdomains exist. Now, what then happens is that through a cascade of events, a messenger molecule eventually gets transmitted via the motors, of course, that can transmit from one part of the cell to another, right to the relevant part of the genome, and in turn influence gene expression. So we've no difficulty in seeing how cells can communicate activities at the level of their surface membrane right the way through to inside the nucleus and to the genome. So I'm now going to ask the question, can any of that kind of process be used by cells and organisms to, as it were, harness stochasticity down at the genetic level? And that's where we come to the focus of Jim Shapiro's book, Evolution, a View from the 21st Century. We go to the next slide, where he writes on page 82 of his book, it's difficult, if not impossible, to find a genome change operator that is truly random in its action within the DNA of a cell where it works. All careful studies of mutagenesis find statistically significant, non-random patterns of change. And genome sequence studies confirm distinct biases in the location of different mobile genetic elements. And then he goes on in the next slide, page 95 of his book, to note that very interestingly, the 2001 Nature report of the draft human genome contained two important figures illustrating what we see when we look at the two sequences for different organisms for transcription factor proteins and chromatin binding proteins. And the answer, which I'll show in a moment, is that it cannot have been the case that those proteins evolved just with one amino acid change at a time. Instead, it looks as though whole domains of those proteins have shifted around and accreted as domains that have been reorganized in the course of evolution. If we go to the next slide, this will show a slightly redrawn version of the figure from the 2001 Nature report on the sequencing of the human genome, but it's showing a comparison in the case of, I think these are chromatin proteins, comparison between the sequences. Those various symbols are just different domains. The domains with the same shape and the same color are, of course, the same domain. And what I'm going to do now, if we press the animation buttons in sequence, we're just highlighting with stars the domains that have clearly shifted in the course of evolution. Now we need to go back to that, that's right, we can hold it there, that have simply shifted around in the course of evolution. I see this as a way in which evolution must have speeded up the evolution of proteins, because I sometimes in lectures to a general audience, I like to imagine two children given forms of Lego bricks to play with. One child is given the original Lego bricks, which are just the tiny bricks which you have to laboriously put together to form any complex structure, like an arch or a wall or whatever it might be. And to the other child, you give the sort of modern Lego brick toys, where many of such components are pre-fabricated for you. And it's very obvious, of course, that if you ask each child to construct a bridge using the Lego bricks we've given the children, the one with the pre-formed structures will get there fastest. And I think even if they just play almost randomly, and I think that must be how evolution has used that kind of method. Incidentally, shifting whole domains around, of course, has been known for a very long time. The next slide shows Barbara McClintock's work, which, of course, was done originally way back in the 30s and 40s, but not recognized fully until she got the Nobel Prize in 1983. And if we go to animate that slide, she wrote in her Nobel Prize lecture that attention will undoubtedly be centered on the genome, well, it certainly has been in the last 20 years or so, and with greater appreciation of its significance of what she called highly sensitive organ of the cell, and pointed out that the genome was clearly capable of responding to unexpected, unusual environmental stress to restructure the genome. In other words, she found what today we would call mobile genetic elements. I have to say, if you go back and read Barbara McClintock's original articles, you need a certain amount of interpretation, the way we would describe such events today, to understand that's exactly what she's talking about. But I think the Nobel Prize Committee were absolutely right to acknowledge her as the discoverer of this process. She did it, of course, in Indian corn, in the plant. So I now come to the last part of the talk, which is on the next slide, the actual harnessing of stochasticity. And what I'm going to do here is just to ask the question, which is on the next slide, how can stochasticity in mutations be used by organisms functionally? Now, one answer, of course, is that we've known about ways in which the organism can do that in its own lifetime for a very long time. It's done by the somatic hypermutation mechanisms in the immune system. If we go to the next slide, it just comes from a nice review that I've used. I'm just trying to remember the title of the paper, but I don't seem to have it on this slide. Anyway, the fact is that what you find in the case of the use of hypermutation by the immune system is that it restricts the hypermutation, which must be done by switching off the error-correcting mechanism either to some degree or completely, so that you get many of those errors in copying remaining as errors, so that you get new sequences. And what this slide simply shows at the top there is that only in the variable part of the sequence to the immunoglobulin do you see the hypermutation. The rest of the system, which of course gives its functionality as immunoglobulin, is kept constant. So this is targeted somatic hypermutation. Now, it's not restricted to the immune system. If we go to the next slide, this is an example from the work of Jack and his collaborators just 2 or 3 years ago, showing that cells possess specific mechanisms to organize their genome in response to environmental stress, in this case by targeting the changes at ribosomal DNA. And then all of this led me to write an article, just the next slide now, which comes from another one of the Royal Society journals, Interface Focus, concerned with viewing evolution from a viewpoint of physics. That's to take, of course, all the things I said in this lecture about the nature of the comparison between biology and physics, physiology and medicine, and I've got the text myself in small print here, so I'll try and just summarize what it shows. Largely, what I've dealt with so far in this talk, which is that there is clear evidence that stochasticity and in particular the range of stochasticity is an attractor at a cellular level, and that it can be harnessed by various mechanisms, including the two that I've just highlighted in the previous slides. So this led me and a collaborator, the next slide, happens also to be a zoologist, who is my brother, Raymond Noble, to re-examine the whole question of just exactly how blind was the watchmaker. This, of course, is a title, of course, that echoes the famous book by Richard Dawkins, The Blind Watchmaker. It's an interesting fact that Richard, who I know well, he's here at Oxford, although we disagree almost fundamentally on the nature of the evolutionary process. We both know that, and we both respect each other's position. But he actually got part of the way to the answer. In that book, that is the Blind Watchmaker, he highlighted the fact that although it might take a very long time indeed for a monkey typing away on a typewriter to, by chance, happen to hit the keys to form a good English sentence, he actually chose a piece from Shakespeare, methinks it looks like a weasel. I think it's got about 26 characters in it, and he calculated that just tapping away by chance, it would take more than the lifetime of the Earth and the process of evolution on Earth for the monkey to arrive even at a single sentence like that. But then he argued, well, but if the monkey could hold any character, any typed character, letter that it actually got right, it would get there in only about 25 or so goes at trying to get the sentence. But of course, Richard then realized, but wait a minute, how would the monkey know that he'd got the answer right? He'd have to do it, of course, by comparing with what he knows the goal to be, but of course he doesn't know what the goal is, and that's where, as Richard himself admits, the idea falls by the wayside, that evolution couldn't have worked in that way. Well, we argue in that paper that actually, if he'd gone a bit further, he's possibly right. His model might actually work. And of course, the way you do it is to enable as the immune system does, you enable the changes to be made in just a particular part of the genome, and you then use the random changes thrown up to select amongst them ones, as the immune system does, that responds to the challenge. And that's why we use the phrase, was she one-eyed? Our idea, of course, is not that there was some form of cosmic watchmaker, but that organisms themselves are the watchmakers, and they do so in a way which can be best described as just feeling their way forward, using stochasticity to respond to challenge and then selecting amongst the variants that enable the organisms to deal more effectively with the environmental challenge. I just want to finish with two other papers that I will highlight that I've worked on recently, which take these arguments a bit further forward. The next slide highlights this article in the American Physiological Society and the International Union of Physiological Societies journal called simply Physiology, with the title just put out last year, Central Dogma or Central Debate, where I highlight a particular confusion that I think occurs in this area. The confusion is that text at the top. The Weissman's barrier, as now embodied by the central dogma of molecular biology, that's the dogma. Of course, the DNA makes RNA, RNA makes proteins, and nothing goes the other way. And he says, this is an anonymous referee, no one has yet shown that protein sequences can be translated into nucleic acid sequences. Well, I agree entirely with the last part of that statement, and I think all molecular biologists would do so too. But I totally disagree with the idea that the barrier is embodied by the central dogma. I think this is to confuse the molecular level processes, which of course is what the central dogma is dealing with cellular level properties, where I think it's been unambiguously shown now that the barrier is actually very permeable. We've even seen recently that sperm can take up RNAs from the father, in the case of the sperm, of course. Eggs can take up RNAs from the mother, and in the case of sperm, they can even transfer DNA in microsomes to the egg cell and lead to the DNA being incorporated into the egg cell. Whether that is functional or not, of course, is a big question. We don't know the answer to that yet, but I think it's very important to distinguish experiments and concepts concerned with cellular level transmission, which is the Weissman barrier in its original form, and whether it's right or not, with the molecular transmission of information, which of course is what the central dogma is dealing with. And finally, the last slide simply takes these arguments to what I think is their logical conclusion, and the very title of that paper, which appeared appropriately in the journal Chaos by the American Institute of Physics, as its title, Harnessing Stochasticity, How Do Organisms Make Choices? The argument is very simply that by analogy with the way in which the immune system uses stochasticity to generate new sequences, we can postulate, and I don't think we can prove at the moment, that organisms might do the same with stochasticity shown at the level of membranes and the nervous system in order to arrive at the ability to make choices. So that's the end of my presentation, and once again, sorry that I can't be there in person. If it's possible for me to hear any questions or for somebody to transmit a question, I'm very happy to try to do that. And if I can be kept online, I'm very happy to listen in to the roundtable discussion that follows this lecture. Thank you all very much indeed.
Thank you very much, Denis. Let me turn the laptop so that we might be able to see the audience. Can you see somebody? Anybody?
Oh, yes, indeed. Absolutely.
Okay.
Yes.
Okay.
Very clearly.
Biologist's
Yeah.
question forward. Anybody? Yes. Can you connect?
Thank you very much for this very inspiring presentation. I am Jean-Pierre Changeux from the Pasteur Institute. My question is, was not Darwin himself able to harness stochasticity, in the sense that already in his theory of evolution, variation was considered as essential in evolution, and why not also generalize the principle to even the development of the organism and of its nervous system as well?
Could you hear that, Denis?
I could hear, but I couldn't interpret because I'm afraid the acoustics distort the sound for me a bit. If somebody can repeat?
Yeah, he will repeat, and I try to make it a little louder too.
I think if somebody could just sum up what the point is, I can try and deal with it. Very sorry about this.
Very, very short. Did Darwin himself, in his theory of evolution, harness stochasticity already.
Yes. Did Darwin, if I got this right, did Darwin himself refer to this kind of process?
Yes. Yes.
In one sense, have I got the question right? Yes. Yes, in one sense, I think he did. His theory, of course, of genules strongly resembles what we now know is happening when exosomes get transmitted to the next generation. Of course, Darwin, in various places in his Origin of Species, struggles to understand what he seems to have accepted from Lamarck, which is that there is at least some forms of inheritance of acquired characteristics. This, of course, was what Weizmann tried to exclude with his concept of the barrier. So I would say that Darwin did actually have this concept. Whether he had the concept of stochasticity as such, I'm not at all sure. I can't find clear evidence for that in his writing, but it does seem to me that he anticipated with his genules idea what we now refer to as the microvesicles that can transmit RNAs, DNAs and protein from one generation to another. What remains open, I think, is to what extent that process is really used functionally, and I think the answer to that is still open to further experimentation. I hope I've got the point right.
Pierre is nodding, so I think that's... So one more question.
Well, I think you know Pierre Teilhard de Chardin. He pointed out that life evolution goes increasing complexity. What do you think about it?
Again, the sound is a bit distorted, so if somebody can transcribe the question, it would be very helpful.
Oh, okay. So what do you think about that?
Pierre Teilhard de Chardin.
Oh God. Pierre de Chardin?
Pierre Teilhard de Chardin.
Okay, I don't know. You tell me about my accent and then you do this. Now, if complexity...
The evolution is in the increasing complexity of life. The Jesuit approach to evolution.
If the evolution is in the complexity...
Evolution. A Jesuit explains...
Are you doing? No, that's not. No, that doesn't make sense. Yeah.
Denis, here's Conrad. We have met in Stockholm. And my question is, my point, Erwin Schrödinger has done much more than that, but not completed in the sense of Einstein. Erwin Schrödinger put what is life on the physical law of entropy, repeatedly. And Francis Crick very much, he wrote him when they had it, that his notion of non-periodic, aperiodic crystals would be very well taken, and it's on the basis of law of entropy, and this law of entropy was the whole basis of all of Einstein's statistical physics. So it was a real basis, and Schrödinger and Einstein were close. So couldn't it be that the law of entropy as a basis of uncertainty, of stochasticity, is actually a law and not just an irregularity? And it's not a complex law, it's not easy to understand, but it's a simple law. And then the idea of complexity is more related to ignorance of the possibilities of entropy. If you look at it at a close, what would you share or have sympathy with such an interpretation of Schrödinger?
Dennis, are you still there actually? I think the connection was. I don't think that was intentional. Although I think sometimes I really want to do that. But this was not intentional. I'm sorry. I'm bringing back online for the session. I don't do this now, right now, and I bring it back up. But we have a short break now, I suggest, until we